Next up, we have Isabel Beerman. She is the Stadman investigator at the NIA, and her work focuses on the age-related changes in the hematological compartment. Around 2015, I guess, there was a paper published in Science that talked about the number of limited productive clones in the hematopoietic compartment, and that caught everyone's imagination. That was one of the most discussed papers, and with all the billionaires and millionaires going to researchers asking, okay, how many clones do I have in my hematopoietic compartment? How do I enhance the number of these? So her talk addresses all these issues and also some of the therapeutic intervention possibilities that she has.
Thank you for that really lovely introduction. I don't know if I can address all of them, but I will try to address some of these issues. And thank you for this invitation to share some of the work. So I'm going to do a slight pivot. We're going to go back to aging. So I am at the National Institute of Aging, so a lot of our work does focus on the far end of the spectrum, not in the development. And as mentioned earlier in talks, aging is a relatively distinguishable phenotype, right? We can look around and we kind of judge aging relatively easily, but biologically it's a little bit more difficult to define. And I think the best definition that we've come to in the aging field is that it's the loss of ability to maintain homeostasis, and it's the inability to repair things once things are broken. And to us, as stem cell biologists, that's sort of a key feature, saying that maybe stem cells hold the key to some of these aging phenotypes. So just as a brief introduction, I think it's pretty exciting that almost all of the systems in our body have now defined adult stem cells. So we have these cells that have the ability to self-renew throughout the lifespan of the organism and are there to repair and to recognize damage and to maintain homeostasis. We just heard beautifully about neural stem cells. Yesterday we heard about skin stem cells. On Tuesday, we heard a little bit about my favorite stem cell, the hematopoietic stem cell. Unfortunately, the fat stem cell is around, but all of these different stem cells have sort of a different capacity. One in the number of cells that they need to differentiate into, and also how much work they need to do to sort of maintain things. So the intestinal stem cells and the blood stem cells are really these powerful workhorses in the sense that these systems have a very high turnover. And as Shiri mentioned yesterday, we have about 330 billion cells that are replaced daily. That's a huge number of cells. And so just as a slight addition to her beautiful comment, so every approximately 100 days, we have like an entire body of turnover of cells. And the vast majority of these cells that are turned over are those in the hematopoietic system. 12% are from the gut, and that small fraction that's remaining are the other stem cells. So we're really interested in understanding what are these changes that occur in the hematopoietic stem cells, given this immense burden that they have. Part of the reason why they have this immense burden is they have a very large repertoire of cells that they're responsible for. So we like to say that the stem cell sits at the top of the hematopoietic hierarchy, and they're largely a quiescent population. So they're maintained in this state that protects them, but when called upon, they'll differentiate and essentially be able to derive all of these different cell types that make up our blood and immune system. So throughout my talk, I'll largely be talking about myeloid cells and lymphoid cells, so it's our adaptive system and our innate immune system, but really the hematopoietic stem cells are responsible for all of this. So what happens during aging in humans? So we have this overall diminished regenerative potential, and mostly we also have this reduced cellularity in the bones. So what happens in humans is called bone marrow yellowing. So essentially our marrow becomes filled with fat, and our productive red marrow decreases. And this happens to all of us, and it's very common in humans, a little less common in our rodent models that we follow. We also know that there's this reduced competence of the adaptive immune system. This has come about a lot recently, talking about COVID and our ability to respond to vaccines as we become older. So part of this is because we have a reduced thymus, we have thymic involution, but it also starts at the stem cell and early progenitor cells, where we have this choice, where we have a reduced production of these B cell progenitors and the choice to go more towards myeloid differentiation. There's this increase in anemia in the elderly, and we see a little bit of a sex difference here in humans. And as what Jyoti had mentioned earlier, we also have this loss of clonal diversity. And this has recently been a very hot topic in human hematopoiesis, because we know that every person, once we get to about the age of 60, we start having what's called clonal hematopoiesis. We have dominance of a few clones that have acquired selective mutations, and these are mainly the clones that are contributing to our overall hematopoiesis. And we also know that this is associated with a sharp increase in disease. And so this is largely myelodysplastic syndromes and myelogenic leukemias, but really at about the 6th decade of life, which is around the time where I mentioned we have this clonal hematopoiesis, we have this sharp incidence of disease. So again, sort of the foundation of the work that we do in our group as stem cell biologists in aging is we really want to know to what extent are these changes we see in the overall system attributable to changes in the stem cell compartment, and what can we do to sort of fix that? And if we fix that stem cell, would we be able to then fix the overall system? And one thing that's sort of been a prevalent theme throughout these talks that we've been hearing is how can these be used for therapeutics? How can stem cells be used for therapeutics? And the one nice thing about hematopoietic stem cells is that this is one of the best recognized form of a therapeutic. So the stem cell is the cell that's the therapeutic of a stem cell transplant, or of a bone marrow transplant, excuse me. And it has been used for a very long time for cancer treatments where you myeloablate the host and a single stem cell can then reconstitute. So we know that there's substantial benefits that can be gleaned from the stem cell population, but we know that one of the largest challenges is the age of the donor significantly influences the success rate of the bone marrow transplant. So we know that something's going wrong with these stem cells as they get older. So a lot of the work that we've been focused on is trying to understand what are these changes that are intrinsic to these aged stem cell populations. So as the unit of transplant, one of the first studies that I was involved with, we looked to see if you transplant the same number of adult stem cells from a young donor and an old donor and put them into a lethally irradiated host, what happens? How well can these old cells compete against the young cells in a young environment? And what we see is young cells do a really great job. They're very good at making both myeloid and B cells, so both the innate and adaptive system, and they do this at about a 50-50 ratio. These old stem cells, not only do they not home and engraft as well, so there's a very small frequency of these shown here as the old donor at 3%, the cells that they make are almost all myeloid biased, so they're not interested in making these B and T cells at all. And we've seen this consistently, and this is a very well-reproduced phenotype, and we see this in humans as well. So in the mouse model, which is what we use, we also see what's called this clonal dominance. So as I explained, we have this expansion of clones in humans, but we also see something similar in the mouse model. So even though we have less productive hematopoiesis, surprisingly, we have a significant increase in the number of these stem cells. So there's many more of them, they're just not very good at what they do. And so what we can see is our immunophenotypic HSCs, which we here by FACS sorting define as 34 negative FLIC2 negative cells, they largely expand, you can see in that second panel in the middle. So it goes from about a 10% frequency, and we have about 2,000 of these cells in young mice, to almost 74% of this population of primitive cells, essentially making them almost 10-fold greater in number. So we have this large expansion of number, but their function goes down. Also what we saw is that using an additional marker called CD150, we could subfractionate this group of stem cells. And it's a heterogeneous population, and we found that some of these cells really like to give rise to myeloid cells, and they're okay giving rise to lymphoid cells. And we have a population that's very happy giving rise to both. In the young animals, the vast majority of the stem cells are the ones that like to give rise to both. So they're balanced, they're great at what they do, they're very good at making both lineages. With age, what happens is we have this very large expansion of the cells that like to give rise to myeloid cells. So we believe that this is also contributing to these phenotypes we see, where we have less B cell output. Earlier, we had a lot of talks about DNA damage, and we definitely see this in our compartment. What we see in aged HSCs is accumulation of these gross mutations of strand breaks in the old hematopoietic stem cells. What we think happens is because they're a quiescent population, they're sort of sitting in dormancy. They are accumulating mutations just because of the environment that's around them, and they slowly over time, have more and more of these mutations. However, what we found is if you take these cells and you stimulate them, you require them to do something, and they enter into cycle, they're actually relatively good at recognizing this damage and repairing it. So their repair mechanisms are still intact, and these cells are able to function. So we have hope that even though there's accumulation of this damage, there's still the ability of them to recognize this repair and fix it. And so I think yesterday there were these comments about bloodletting. I remember my previous advisor used to say, maybe the ancients had it right with the bloodletting, because you stimulate your stem cells every once in a while to kind of come out of quiescence and fix things. You won't have this massive accumulation of damage. It's like in my, I'm embarrassed to say, it's like in my apartment. Like if I'm at steady state, it's kind of a big mess all the time. So it takes a really long time for me to clean things up and make it presentable when I have guests. But if I was constantly cleaning things, it would be a much less burden, and it would probably be done a little bit better. So we're thinking that maybe slight stimulation of the stem cell population could be a good thing as well. And then finally, this is sort of what we're building my current lab on, is that we found that there's an altered epigenetic landscape. So during aging, it's typically associated in differentiated cells with this loss of DNA methylation. And this is thought maybe because it's epigenetic drift. There's loss of maintenance of these epigenetic marks, and so when you start at a high level, you'll obviously decrease and become hypomethylated. But interestingly, what we found in our hematopoietic stem cells in the mice is that even though they lose function, what happens is that they actually become hypermethylated. And this was reproduced by other groups. We found this to be quite surprising, and we think that it's something that must be concerted, because it's not something that could happen randomly. And we do see the consistent changes if we force these cells to cycle excessively. So we can force them to, by transplant or by 5FU, to overcycle. So there's this delicate balance where we want some cycling, but not too much cycling, because then you have these marks that then we believe contribute to some of these altered functions. So this is kind of what my group now at the NIA focuses on. So we know that these are sort of the functional changes that we see. We really want to understand what's underlying these functional changes. What can we do to tweak them to hopefully reset the functional potential, not just for us as we're aging, but also as an increase of the therapeutic pool that we could use of donors who would be happy for bone marrow transplants. So the story I want to tell you about today was led by a really fantastic postdoc fellow, Lei Zhang, on the left, and a bioinformatician, Bong-Soo Park, who has really put together the story that I'm going to be telling you today. And what we wanted to look at is, in the stem cell compartment, it has been reported not only by us, but by other people, that there are very significant transcriptional changes that are associated with aging, and they happen very consistently. So we wanted to know, what are the epigenetic drivers that cause this? So we did ChIP-seq, ATAC-seq, and HiC. And the one thing I want to highlight is there have been a few publications before looking at this. But as I mentioned, in the young mice, you can only get about 2,000 of these purified cells. So it's really challenging to do things at a very clean and reproducible level for HiC and for ChIP-seq. So it was really due to this outstanding work by my postdoc Lei Zhang, who has managed to get really high quality reproducible data on these very small numbers and many animals required for doing these experiments. So the first thing I just want to show is the reproducibility of the transcriptional changes. So when we look at what's being expressed in young HSCs and old HSCs, even though they do the same function, they still are able to reproduce all of the cells in the hematopoietic system. We have a very strong separation between their transcriptional profiles. And interestingly, we have almost a relatively even balance of the number of cells that are, number of transcripts that are upregulated and downregulated. So around 1,500 that become upregulated, and a little bit less that lose expression. When we look at these genes and how they might actually contribute to the functional changes of what we see, it quite nicely correlates with the functional changes. So genes that become downregulated in old HSCs are really important for the differentiation of lymphocytes. So again, the lymphoid lineage, these genes become repressed. And the upregulated genes really have to do with cell mobility and migration, and we haven't quite figured out how that associates with their functional changes, but it's something that we consistently see. And we use this nice, what's called a cell radar plot, which looks at genes that are important in all of these differentiated cell types. So starting at the stem cells down to differentiated B cells and progenitor cells, in blue are genes that are more robustly expressed in young. And I'm sorry if you're unable to see this, but kind of the lip of this butterfly plot kind of points to genes that are involved in B cell production and lymphoid production that are highly expressed in young cells, again, promoting their differentiation to the lymphoid lineage. And in the old stem cells, what we have is sort of these programs that are important for self-renewal as well as myeloid differentiation. Again, very nicely fitting with this change in what they actually do, but not really informing us how this is happening. And so this is how we get involved with epigenetics. And how I like to think about epigenetics is, I don't know if you guys did this, but during COVID, I watched a lot of the British baking show. And the second episode, or this, their second task is always, here are some ingredients, bake this amazing thing. So it's essentially like what our DNA is without the instructions. So the epigenetics I think of as sort of the instructions needed to bake a cake versus, pancake. Or, I'm not a very good baker, but it always looks amazing for when they do it. So the epigenetics essentially tells your skin cell to be a skin cell, or your brain cell, your neural stem cell to be different than your hair. So we think this is really important, and what's most intriguing to us as a therapeutic is it's something that's modifiable. It's inherently modifiable. We don't need to go in, we don't need to change the DNA, we just change the instructions that's given. Not the ingredients, just the instructions. So the first thing I want to talk to you about are histone modifications. And so the main ones that we looked at are a permissive mark, H3K4 trimethylation, and usually this mark allows for active transcription, and you see it at both the transcription start site and sometimes near the enhancer regions. We also looked at K36 trimethylation, and this is a mark on the histones that's during active transcription, so you know that it's actively being transcribed when we have this mark. And we also looked at a repressive mark, H3K27 trimethylation, and again, this is found at both the promoter region and in the body, and it prevents the genes from being expressed. We also looked at K27 acetylation, and this is sort of a poised mark, usually found at enhancer regions. And when we compared these between young and old HSCs, what we found is that in most of these globally, looking at either the transcription start site for H3K4 trimethylation, the permissive mark, we see this overall decrease in this mark in old HSCs. We also see a decrease in K27 trimethylation and also K36 trimethylation. So globally, we see that all of the marks, or these three marks that we looked at, were globally decreased. However, the poised mark, the K27 acetylation for enhancers, was at the same level. And I know it was several slides ago, but one thing to remember is that surprisingly, we had more increased expression with aging. So even though all of these marks decrease, so our permissive and active marks decreased, we still have increased transcription. So given the resolution that we had in this data that we generated, we were able to go in and say, in these regions that we have differential transcription, do we have changes in histone modifications that may be driving this? So we used a slightly more stringent criteria for what we said was upregulated and downregulated, and looking at 738 transcripts that were upregulated consistently in old HSCs, we found that most of what happens is this loss of K27. So it's the loss of repressive mark, we start turning on genes that maybe shouldn't be expressed in the hematopoietic stem cell. When we look at genes that lose expression, it's mostly due to a loss of K4 trimethylation, this permissive mark, but we also do see the slight increase in K27. So it's not suggestive that it's completely just this loss of maintenance, but it's something that's being actively added in some places, because globally we have loss, but we're finding these sites that are being actively turned off by this increase of K27 repression. And we're also very curious about these transcripts that didn't change. So do we have histone changes that are occurring that have no relevance to transcriptional changes? Oh, wow. So the bottom doesn't make any sense. But essentially what we want to say is all the marks that we looked at, surprisingly, the K27, this is, excuse me, so the first mark is K4 trimethylation, so we have this decrease in the permissive mark. And we also have a decrease in K36. So the active and the permissive marks are both decreased, yet we have the same levels of expression. So we know that although these epigenetic marks are contributing to some of these transcriptional changes, it's not the only thing. So what we believe is happening is that there's some sort of change in how this RNA is either being degraded or maintained, but we have yet to kind of establish what it is. So even though we think epigenetics is very important, it's not the only answer. So one thing that's unique, I would say, about stem cells, and it's really been important in the embryonic stem cell field, is our bivalency marks. So bivalency is sort of a way of cells to poise themselves, to be ready to be at the starting blocks and be able to both go and stop. And so they have the K27 trimethylation as well as the K4 trimethylation, so both the repressive and the permissive marks. So I really like this sailboat analogy. So it's like an anchored boat with a sail up. So if you remove the anchor, you just take off. Or you put the sail down, you know you're staying in one place. So it's really these poised marks that allow for stem cells to quickly make decisions of whether they want to go and differentiate into one lineage or another. So what we found is that during aging, we have maintenance of many of these bivalent promoters in both the young and the old, but there's also very unique bivalent promoters. So you lose some of these bivalent promoters with aging, and you also gain some bivalent promoters. So the vast majority of changes that we see at the promoter regions for K4 trimethylation or the repressive mark K27, almost half of them really occur at bivalent promoters. So we think that this affects the lineage potential choices of the stem cells as they're getting older. So we wanted to examine some of these bivalent changes, and this is just a nice plot of one showing how nice our data looks, and also to show this is a bivalent promoter at this gene called OSMR. This is an important gene for megakaryocyte and erythroid development. And what you can see is in young, you have both the marks of K4 trimethylation and K27 at the promoter region. So the gene is actually going to the right. With age, what happens is you lose that K27 mark, and you have this only active and permissive mark. And what you can see, I'm sorry with the pointer, what you can see at the bottom is that we now have robust expression of this gene only in the old HSCs. And one of the phenotypes of old HSCs is that they have sort of this hijacking method to go directly into megakaryocytes. So we believe that one of the reasons why they're able to do this better than the young HSCs is because we've lost this repressive mark of K27. So again, we have this very nice correlation between epigenetic modifications that consistently happen in old HSCs and functional potential. When we looked at these changes in bivalency, what mainly happens? So we have a lot of upregulated genes, some downregulated genes, and a lot that don't have changes in expression. So to us, we think that that's interesting, because in the stem cell, again, these are poised markers for differentiation. They're not important for the stem cell themselves. They're going to be important when they make the decisions for differentiation. So we're very curious in following up on these genes that lose bivalency but don't have transcriptional changes. The vast majority of these changes, though, that are associated with transcriptional changes are really due to the loss of the repressive mark K27 trimethylation. So now that I've sort of explained we have these changes in histone modifications, there's other marks that we can look at as well. So these are regions that are already open, and they're accessible chromatin regions where transcription factors bind, and the histone modifications are kind of subsets of these regions. So here you can see just a TAC-seq profile, and essentially what we're looking for are these regions that are open in young and old HSCs. Again, very similar to what I showed you with the transcriptional profile, just the chromatin organization and the accessibility of the DNA in old HSCs is very different than what we see in young. And surprisingly, what happens in these old HSCs is we have a lot more open chromatin. So there's a lot more access, and there's a lot more of these regions that should be closed and regulated that are now open in the old HSCs. Surprisingly, we also see these regions that become more closed, and they're very consistent throughout our multiple replicates. When we looked at where these regions of this changed accessibility are, as a geneticist, we're always most excited about regions that are promoter regions. Those are the most important regions. These differentially accessible regions are really far away from what we would call like the meat. They're not near the transcription start sites. They're in either intergenic or intronic regions. So we thought, well, what else is out there in these intergenic regions? And this was nicely brought up by Andre on Tuesday when we was talking about these transposable elements that kind of live out in these regions. So these transposable elements that you don't really want hopping around your genome are usually protected, and they're in these regions that are repressed by epigenetic modifications. What we see during aging in our old stem cells is that these regions that should be closed now become more accessible, and we have increased expression of these lines, the signs, and also the LTRs. So we have this increased expression of these transposable elements that could also be causing a little bit of chaos in these old stem cells because we've just lost this regulation that should be keeping them from hopping around. What we also found is that even though there weren't a huge number of these regions that were in our promoter exciting regions, the regions that we were enriched for are again very functionally relevant transcription factor binding sites. So regions that gained accessibility in the old HSCs have to do with, were enriched for BATF and MACE1. MACE1 is a very important transcription factor for HSCs. My previous lab had shown that it's one of the factors required for reprogramming back to an induced HSC. So it's important for self-renewal potential, and BATF1 is also very important in both self-renewal and in the differentiation choices. And intriguingly, for the regions that lost accessibility, SPI1 was, you can see outstandingly, the most highly enriched transcription factor binding. This is really important for the regulation of these inflammatory factors. In the aging field, it's very common, and one of the biggest drivers, we believe, of some of these aging phenotypes is this inflammaging. And SPI1 is one of the regulators in the hematopoietic system that controls this. So we're now having this loss of accessibility of the regulation of these inflammatory signals. So now, one more big thing that we were really excited about that people hadn't done before is looking at the chromatin organization. So there's about 2 meters of DNA that we pack into our cells. And so this is done by really this immense chromatin compaction, and it's very difficult to analyze. So we were very excited to get this view of what's happening in the compaction of this chromosome or the chromatin organization between the young and the old HSCs. And I won't go into detail, but the main idea is that the big compartment, or the big sections, are called compartments, and these smaller interactions are called TADs. So what we found in the old HSCs compared to the young HSCs is in these long distance interactions, there was less touching. They had less interactions at long range, but interestingly, these short range interactions happened more frequently than they did in the young. And this is a little counterintuitive to some of the other publications that have looked at this in different cell types. So we saw a difference in that there's a decrease essentially in this long range and increase in short range. So we wanted to look at this a little bit more in depth. So I'm showing you here this interaction plot on chromosome 19, one of the small chromosomes. And what I hope you can appreciate is on the diagonal, we have a lot of red. And so that means very close interactions in the old HSCs are happening more often than they are in the young. And what you can see kind of on the far ends is that there's more blue, meaning that in young, we have more of these long distance interactions that are happening. Again, very much validating what we saw I just showed you in the line plot. And the difference between the A and the B compartments is that the B compartments are largely considered to be quiet compartments. They're usually heterochromatin, whereas the A compartments are usually these compartments that are more active and have active transcription happening. When we zoom into these regions that are more touching in the young compared to in the old, what we see is that it's largely in these inactive compartments. So in the young, they're closed and they're close together with the B compartments. And then what happens in aging is they just become, we like to call it loosey-goosey in the old HSCs. And this was done really in a collaboration with KG Zhao's lab and Yongqing Zhao at NHLBI, who helped us with this analysis. Again, the changes that occur are mostly all of them leading to decreased contact in the old HSCs and this BB interaction. And then when we look within these TADs, so these smaller range interactions, they're almost all increases in interactions. And surprisingly, these are also happening at regions that we would associate with heterochromatin. So there are these regions, if you can see on the far right, that are typically associated with high levels of K27 trimethylation. So the fourth panel down, the blue H3K27 panel is showing that it's sort of closed in the young. And then you lose that K27 trimethylation in the old, allowing it to be more open. And also this is replaced by K27 acetylation, which is a poised marker for enhancer regions. Oh, I think the standing means my time is almost up. I'm sorry. So this also correlates very nicely with showing that even though almost all of the changes we see in histone modifications are this decrease, what we do see is a significant increase in our K27 acetylation at regions that are potential enhancers in the old. So we're saying that in these regions that are usually closed off in the young, we now have this opening, and we now have these new interactions of enhancer regions with potential promoter regions that would allow for aberrant transcription that's happening in the old HSCs. So as my final slide, one of the beautiful things that we are able to do at the National Institute on Aging are intervention studies. So we were very excited to get involved in one of the intervention studies of caloric restriction. And what let me get to this. So in these aging interventions, so what we did is we calorically restricted mice, unfortunately for the lifespan of these animals. So for 2 years, these little mice got a single pellet of food throughout the lifespan. They were much healthier, they were much more active, they maintained a lower body weight. And when we looked at the hematopoietic stem cells from these mice that had been calorically restricted for their life, we could see that in some of these regions, and I apologize for the smallness of this, that you can see an intermediate change. So if you just look at the top panel, this gene P-selectin has very low expression of K4 trimethylation. And in old HSCs, we have a really high level of K4 trimethylation, so it becomes very active in old HSCs. With caloric restriction, we now have a mitigation in this amount of K4 trimethylation and also a reduction in the expression of this gene. So essentially what we're looking at is how do these interventions affect the epigenetic profile of these cells, and how can they be used to sort of, what are the key things that change, and are there ways for us to modify them without having to go through the challenges of caloric restriction throughout our life? So with that, I'd really like to thank my team for all the hard work that they've put into it, and thank you for your attention and the invitation.